Yeah, if I was going to give a very broad, basic title for what evolutionary hologenomics is, and this is what I do in talks all the time, it's really re-evaluating life through the hologenomic window. But if you want to paraphrase Dr. Spock, it's basically, it's life, Jim, but not as we know it. And this is really actually key to some degree about what we're doing. Our aim is to look at some of the big questions in life that you will have heard about if you've studied biology at all, high school or at a university or so on, especially in ecology and evolution. But while there are many accepted paradigms for how life works, we have a slightly different opinion that I don't think is heretical, but other people do. And that's really what is at the core of our center. So the aims of my brief intro now are to introduce the Holobiont and the hologenome, what these terms mean, give you the basic ideas for considering if the Holobiont could change our view on life's progresses. So in 2013, I actually was talking to this man here. This is Gary Graves. Gary is was the curator of birds at the Smithsonian. He was apparently Carsten Rabeck's PhD supervisor. He's a really interesting old guy from the US, and he basically cornered me. He was in Copenhagen and he trapped me, and he knew I had DNA stuff I could do. And he was like, Tom, have you ever thought about how a vulture can eat a rotting carcass and not die? And to be honest, I never had thought about that question, right? I mean, I knew they did it, but I hadn't thought about it. And Gary was like, wouldn't it be awesome to investigate that? And I was like, well, I guess so. And then Gary was like, you know what, I have this unique opportunity. And so he said they were going to go into Nashville and they were going to kill a load of black and king vultures because they're a problem. You know, they crap all over your car and stuff and they destroy your roof and whatever, but they're protected. And he said he was going to go with them and he could arrange it so he could actually individually euthanize every single vulture and then take all sorts of samples. And what he wanted to do was basically get genetic material for the bird, but also swab the face and swab the gullet and swab the stomach and swab the intestine and swab the legs and get all these swabs. Because he was like, don't you think it's fascinating? They're eating this really nasty, rotting, toxic crap and they're not dying. You know, what's going on with the microbes? And Liz Zepeda, who some of you will remember was a PhD student here, she basically took a load of shotgun sequence data that we generated on the microbes and she crunched it. And she basically found out the microbiome in the guts of the vultures were full of all sorts of microbes and microbial genes that might actually help them deal with the diet. There were, for example, predatory eukaryotes. There was microbes that secrete things to stop colonization. There were phages, there were beneficial bacteria and so on. And I found this kind of interesting because basically what we were seeing is what in retrospect could be called a holobiotic basis for adaptation to rotting carrion. And it's very, very simple. The point is, if you want to adapt to the niche of dealing with nasty rotting roadkill, you don't just need the adaptations in the vulture itself, but you need these complementary microbes and you need the union of the two to fill the niche. And this is really what the term holobiont refers to. It's the idea that on its own, an organism cannot fill a niche often without the help of its microbes. So if you want to understand life, you can't just look at the host. You need the host and the microbes or you're missing the picture. And so the point is that the microbiome frequently complements the genome, in particular the gut microbiome, but also other bits. And really, if we want to study how life works, and I mean the organismal biologists, we cannot ignore the microbes. So if you want to ask, how does your koala live somehow on those weird leaves that it gets stoned on, or how does the panda eat plants, although that's an interesting example we'll get back to. How do vampire bats drink blood exclusively? And actually this, on the one hand, this was quite striking to me. On the other hand, to anybody who's ever worked in the agri-sciences, this is obvious, right? Because everyone's known for decades, probably centuries, the only reason cows and other ruminants can eat grass is because of the microbes in them, but I was kind of missing that window. And so to get to the terms, the holobiont is simply not just the organism, but the organism and its microbes. And a hologenome is the genetic content of the two. This term's actually quite old. It was first proposed by Margulies, who many of you will know was the one who came up with the symbiotic hypothesis that mitochondria were basically bacteria encapsulated by bigger life forms to create eukaryotic cells. These terms are problematic, and we may get back to that, but Holobiont, studying life as more than just the organism, but its microbes, and the hologenome is studying both bits at once. Now, as I said at the beginning, this is quite a good term because it's a very vague term. To be honest, if you do anything where there's genomics and microbes, or even one of the two, you're kind of doing hologenomics as long as you're thinking about the bigger picture. So let's jump forward again to quite recent history. Again, about 2014, I got talking to Morten and Anton separately. Through the discussion, we got discussing, okay, what could we do beyond our regular animal systems that we do? And so basically, this was driven by the fact that I'd worked at that point on vultures and vampire bats. And of course, at this time, I was reading more and more about the crazy, crazy reports you might have heard about how microbes do all sorts of amazing phenotypic things. They're very, very hot in health right now. There's pretty much no disease right now that people aren't somehow listing as being driven by the microbiome, whether it's obesity or schizophrenia or IBD or arthritis or so on. And so, we got talking about how can we stick these things together. And about that time, there was some very interesting papers coming out about, again, phenotypic effects of microbes on non-human things. So, for example, studies reporting how what animals can eat is very much conditioned by their microbiome. And I'm talking about at the within species level here, right? For example, number B here, different populations of these wood rats, I think they are, either die or don't die, depending on what they eat, depending on the microbe inside them. There is, as many people know, my favorite study ever that came out about hibernation in brown bears, where this team of bold Swedish people sequenced the microbiome of the lower intestine of brown bears that were active and hibernating. And they found that the microbes are actually driving to some degree the hibernation process in the brown bears. They're actually leading to metabolic changes. They did that by actually doing fecal transplants into mice using what they found. As I've told many people, the crazy thing about this is how they sampled the hibernating brown bears. It's unclear. They don't say that, but a lot of respect to them for that. And in fact, there were more and more things coming out. How do animals regulate to different temperatures? Behavioral stuff, crazy stuff on behavior. Whether, for example, mice are happy to be exposed in the middle of a room or not, or jump off things or not, is conditioned by the microbiome. And this really got us thinking, and we essentially, to cut things to the chase, came up with the idea, maybe the Holobiont, the union of the microbe and its host, is a key unit for explaining some evolutionary and ecological processes. So basically stop focusing on the individual parts, which is what I was guilty of doing for 15 years. This is a very controversial statement, and just for the record, because there are a couple of people here who've actually been trained in evolution, this is not the same as saying the Holobiont is under natural selection or not. I actually don't care very much about that answer. It's interesting, but it's not there. But the point is, I think if we want to study ecology and evolution and explain certain things, if we don't look at it from the union of the two, we're missing the point. Why is it? Well, actually, there's a very simple answer. This is a figure I think Anton made for one of the papers. The metagenome, the microbiome, gives you increased flexibility. Now, what do I mean by that? Well, if you think about regular genetics, right, you will have in a population some genetic variation. And if the environment changes, the dogma is, well, yes, you can select on genetic variants to then bring the mutants that you have in you to dominate, to help you adapt to the future, which is great, but that's a very, very slow process. If you have a really important mutant in a genome, for example, the mutant that allows Europeans or Western Europeans to drink milk, that didn't go from 0% or 0.1% in the population to 70% in the population overnight. That took generations and generations and thousands of years. And the question is, can adaptation really happen that slowly if it's going to be beneficial? The beauty of the microbiome, you've got inside you this enormous catalog of microbes full of genes that do stuff, and you can change them instantaneously. So it's a really rapid mechanism for adapting. You can adapt within weeks if the right microbes get in there. A very good case in point, let's say you, and sorry, Sham, this is not in any way to be stereotypical, but let's say I go to India and start eating street food. I'll probably have a bad stomach for a number of weeks, right? Eventually I won't, probably because my microbes have changed and they're helping me adapt to whatever's different in the diet, right? So that's helped me recover. My genetics did nothing really at that level. It was that rapid change of the microbes. So they give you increased flexibility. So with these thoughts in mind, we, and I say we being me, Morten, Anton, and a few others, thought, well, hey, maybe we can take this idea, which doesn't really seem to be discussed. Yes, people are looking at what single effects can microbes have, but nobody's thinking about it in the bigger picture, ecology and evolution. Maybe we can go to the Grundforskning Fund, the Danish National Research Foundation. And the team at the time is these wonderful people, myself. And the reason I went to these people, I needed a very strong molecular biologist. So I went to Anders. I needed some really good, deep thinking biologists. And so Rob and Alina. I needed some people who actually can do math and computation. So Sham and Ida. And then, of course, Anton and Morten, who had sort of birthed these babies with me. And we put the grant in. And we basically said, look, we would like money to study some key questions. And these are really what lie at the heart of the center. So some of the key questions: is the microbiome a phenotype of the genome? What do we mean here? Well, basically, does it matter what your genetic variation is with regards to what microbes do you get? Or do different people's genomes condition them to accept different microbes? This is actually a very fundamental question. It's a very debated question. I absolutely believe it's true. Some very influential people think it's bullshit. Now, actually, it's kind of obvious the answer is yes if you look at the interspecific levels. So if you compare, for example, monogastric animals versus ruminants versus hindgut fermenters, they have very different structures inside them and they have very different microbes. And clearly they have different microbes because their guts are just built differently. So it's not really surprising. But it's much more controversial at the level of the within species, the intraspecific. And why is that? Well, it's because it's really hard to study. So here's an example paper published, 5-6 years ago. And what this study did was they did classic metabarcoding of the microbes in the poo of different human communities. So bottom left, we have Hadza hunter-gatherers in Tanzania. We have on the left side, pastoralists or city communities in different parts of Africa, East Africa and West Africa. And then we have Europeans and Americans. And the gray is basically just a standard baseline network that's the same for everything. What you need to look at is the colored dots. So you will see that, for example, the intensity and the size of the colored dots and which ones there vary differently. So, for example, these big orange ones, the Prevotella in the Malawians and the Burkina Faso, are basically absent in Europeans, right? Now, the question is this genetics or is this just because they have very different diets? And clearly we are living in very different situations and the environment has a huge effect on the microbes. So at about this time, some other teams were trying to get around this by studying twin cohorts. So these are very clever studies. They basically say, well, we need control for the environment and let's assume that twins basically grow up in the same environments. And if you have identical twins, like the Olsen sisters here, or non-identical twins, like the Johansson twins here, at least their parents are the same, their house was the same, their food was the same. So the environment is kind of stabilized. So therefore, if we see differences in the microbiome between Ashley and Kate Olsen or Scarlett and Hunter, that's got to be genetic variation, because bear in mind the non-identical twins have got more variation than the identical ones, which basically have none. And by doing this, they typically find, yes, the microbiome is more similar for the identical twins than non-identical twins. But part of the problem with this is that people say, yes, but it's such a tiny difference, it doesn't mean very much. And also there's no mechanisms. And so what we said was, well, look, if we want to study this, we need to do it properly. We want to build what we call the hologenomic framework. And we said, look, if you really want to study this, you've got to sequence for whatever system you care about, not only the genome of the host and the microbiome within the host, but also everything else that could be important, the transcriptome of the host, what genes are active, the epigenome of the host, what genes are actually turned on, and epigenome and transcriptome and to some degree proteome tie together, the metabolome, which is a union of the both. And again, not just the microbes, but the transcripts in the microbes and the proteins and the metabolites they're producing, because that way you can not just look for a correlation between genome and microbiome, but you can use all these other bits to look for causation and then start to do important stuff. So as part of the center, we said, you know what, let's build this framework for doing this. And I think this is going to be the most powerful thing, having the tools and the capacity to do this, because this can be leveraged for lots of future studies. So once you've got these tools, other questions arise. Under what situations might the Holobiont shape life's processes? Now, this is tricky because there are key questions which are very philosophical. What actually is this Holobiont? What is a hologenome? How are they evolving together? And for example, is the Holobiont your genome and every single microbe living on you at every possible time in your life, or is it just a stable community you're in there or so on? Very, very unclear. Very nice to solve, but I'm not sure there's a black and white answer. Another set of questions: what kind of basic evolutionary or ecological questions could such a relationship explain that are normally explained in a different way? For example, invasion. Could we explain invasive success? Which species are, for example, expanding and which are not, as a combination of the genome and the microbiome, similar to adaptation or competition or even domestication? And similarly, what kind of really big questions might the relationship explain? So I'll give you one of my favorite examples here, a thought experiment. This is one I stumbled across a few years ago, which I found kind of interesting, and it's unexplained. It's the evolution of tetrapod herbivory. So basically, when the first four-legged animals came onto land in the Devonian about 450 million years ago, whenever it was, the land was covered in plants, the land was covered in insects. But what was interesting was when they first crawled out of the water looking like this, it took, they were eating meat basically and insects. It took about 100 million years for them to get around to eating the plants, which is really weird. As you probably will have been taught once if you're a biologist, the basic dogma of evolution is you put an organism in a new environment and the adaptive race starts. Things adapt to fill all the niches, right? So they have their own competitive advantage. For some reason, they didn't bother to eat the plants for 100 million years. It's possible they were too high off the ground and they couldn't get to them. But paleontologists don't really have an explanation for this, right? And it seems pretty striking. And if you, I mean, I guess you could say, well, maybe it just takes 100 million years to evolve a gut that you need to do it. But I think that's probably unlikely. I mean, in mammals, in 100 million years, this rodent-like thing evolves to pretty much the diversity of mammals we see today. So clearly, changing form is not a major issue. What I think is going on is maybe it took 100 million years for the genomes of these early tetrapods to adapt to have a stable relationship to encourage the right microbes to live inside them that you need to make eating plants worthwhile. I think there's a bit of preliminary evidence for this based on pandas. So as you probably know, pandas are one of the most retarded life forms, not quite as retarded as seals or koalas, but they're pretty close. They have to eat plant literally every hour of the day. They eat bamboo constantly. That is because they can't get enough energy out of the bamboo. They're quite capable of eating other things. They will eat lizards and chicken and whatever, but they just for some reason like to eat this really difficult to digest thing. It's kind of like when people do these diets where you just eat cabbage or something, right? I mean, it's not necessarily the best thing for you, but you do it for a reason. Now, when this paper came out in 2015, it was a microbiome paper, sorry, a study of the microbiome of pandas, and they just did 16S sequencing, and basically the white dots are basically pandas, microbiomes. The green are herbivores. The blue and red ones are basically omnivores and carnivores. The key thing is the panda, despite wanting to be a herbivore, has got a carnivorous, omnivorous microbiome. No wonder it can't get any goodness out of the bamboo because it hasn't got the right microbes inside it. Now, I don't know exactly how many million years the panda's been trying to do this, but clearly it should have been more successful. Think about how human form has changed or hominid form has changed in several million years. So it's clear that the stumbling block here is getting this relationship with the microbes, not changing its side of the body. And this is why I think maybe this is what, took this big delay in the evolution of herbivory in the animals. So a different explanation based on the need for the microbes and the difficulty of getting them is what I would hypothesize. Another very big area that, and this is really Rob's specialty, is fermentation. So actually, how do animals get around the ability to do something naturally by actually incorporating microbes on the outside of the body, whether it is to make alcohol or sourdoughs or so on? And Rob will talk a bit about that. So to get towards the end, basically we have what's called 5 themes in the center. This is just a management thing really, but we basically said we would do 5 things. We would generate the full hologenomic approach, which is generate the lab methods and the computational methods to explore these kind of questions. Number 2 is to study the interplay of the genome and the microbiome. Is the genome actually constraining the microbiome and how are the two evolving? Number 3 is to look at the kind of everyday questions in ecology and evolution, adaptation, for example. Number 4 is the big questions. Can we tackle some of these big paradigms, evolution of herbivory, for example? And number 5 is the impact of the hologenome on human ecology and evolution. So for example, how do we exploit microbes on the outside? There is actually a hidden 6th agenda. I say it's hidden because in the original grant we wrote, we basically had exactly the same, but we said, and in addition, we believe that this approach can revolutionize some applied questions. For example, production of animals, chicken or salmon, as some of you know, the Hollow Food Project is basically a spin-off of our first application, and humans. We were actually told to remove that off the record. They thought your aims are too big. I think that was a very incorrect decision by the foundation, because the point is the framework we're building is equally applicable to everything. And that is why I'm quite happy that we are, now we've got the money, we will be exploring both the basic but the applied stuff as well. And really, just to put it into context, I mean, many of you will know about the projects, Hollow Fish, Happy Fish, Hollow Food, Crappy Fish, and so on. But the idea is very simple. If you believe that microbes contribute to phenotype of importance for example, farming. If you want to get bigger salmon, you need to give them the right microbes so they get the right amount of energy out of food. The whole idea behind probiotic treatment and diet manipulation for both humans and animals is let's try and prod the microbiome of an animal to the right microbiome to make them more efficient at digestion. Now, what we say is, but if the microbiome is conditioned by the host genome, you have to do it in this knowledge of what genetic variants in the animal are actually shaping the microbiome. So ideally, if you want to make bigger fish by giving them a probiotic, you need to first of all pick the right genetic background of salmon to be successful. And so that's basically hologenomics. So what skills are needed to achieve this center? Well, actually, it's kind of everything. I mean, we of course need the biologists because they've got the big questions. And I mean both field biologists, but also theoretical biologists and applied biologists. Absolutely need the omics experts. I am not at all an omics expert. I know how to get omics data. I know who to talk to about it. But we want to bring in every possible bit of omics that we can not just see the correlations, but explain the causations. We need molecular biology gurus and geniuses, because what we're trying to do is not at all tricky. One thing that I've stupidly done for 20 years is pick things that are not easy to do in the lab. And there are a lot of challenges. If you talk, for example, to anyone who's tried to work on a salmon microbiome, they'll tell you horror stories. You need big data experts. We're talking about massive amounts of data here. Omics is big. That's basic computation. We need modelers and mathematicians and theoreticians. Absolutely need population genomicists, because at the heart of it, I believe the microbiome is a phenotype, and to study genotype phenotype is just population genomics. Statisticians, because it's very easy to see correlations, very hard to see causations, and some very organized people, which again is sadly not me. So the team, the original squad I already presented, it was Anders, Rob, Alina, Sham, Ida, Anton, and Morten. But luckily, even since getting that, we've had the ability to expand the PIs. So luckily, we managed to persuade Sandra to defect from biology and come and join us. And that's fantastic, because Sandra's pushing the human angle, which I really believe is the future. And I believe we can help a lot there. And we're working very hard to push Astaiska that way as well, because Astaiska is a real animal behavior expert, which I think is an absolutely critical and important area where this will be helping. And actually, I don't think it will stop there. I'm really keen. We are the only place I know in the world studying evolution from the context of hologenomics. And I want us to stay that way. And I think that if we can prove it really works, we will start attracting more and more grants and talent, and we will grow until we are even more uncomfortable space-wise than we are now. Now, of course, in addition to the PIs, there is key support elements. Cecilia is the center administrator, and she will briefly talk in a minute about a few things there. And Christina is our outreach specialist. And I myself am not a huge fan of outreach, but I know it's massively important. And it's really important we do the right outreach for reasons that I'll come back to. Both of them will say something briefly in a minute. There are a few other permanently hired staff. Sarah, who I literally couldn't find any photo on the internet apart from this one. I don't know, Sarah, why you're hiding from the internet. And Ida Borman-Nielsen, they're basically in very, very technical roles, mainly helping out myself and Anders Hansen, who have a lot of admin on our plate to not get too behind. But of course, there is a number of other staff hired directly from the center, a number of PhDs and postdocs, and more to be hired over the next few years. I'm not going to bother giving the names now. We can come back to that. But of course, there are a number of other people here who I would say are thematically linked staff. I was going to change this poster, but frankly, it was just too funny in the original context. Of course, it was going to say, I want you to be in the center. But basically, I really do view the center as a hub, a place to work on hologenomics and expand beyond the original grant. And actually, many of the grants we have now, HoloFish, HoloFood, the Willem Cat grant, Finding Pheno, they are essentially spin-offs of our original application to the Grundforskning Fund. So even though they were awarded maybe contemporarily, we only got them because we started pushing these ideas to the DNRF and so on. So I personally view them as very much within the spirit of the center. And I would argue that anybody who thinks, you know what, I can contribute to this understanding of genomes and microbiomes, or I want to bring this approach to my work, is very welcome to be in the center. And that is why I hope we have 41 or so odd people today, although maybe only 20 or so are directly paid for it. In my view, you are all part of the center. So to end with, let's jump to the future. What do I want to do? Well, basically, I do want us to be the place to study evolutionary hologenomics, which is not very hard right now because a lot of people think we're heretics and we're talking crap, but hopefully they will change and then people will be begging to come and join us. How do we do that? Well, obviously we need to keep the Grundforskning Fund happy, the National Research Foundation. We have a 6-year grant that could become a 10-year grant if they think after 5 years we've done a great job. That is why we have a very organized Cecilia and Christina to help us do good reporting that makes them happy and do lots of outreach to make them happy so people see what we're doing. Of course, by being successes and growing really helps. So for example, just by bringing on Sandra, I know Sandra, you think I'm being nice to help you get a job, but you're being even nicer to me because that's already a big success. I can show, look, we're attracting and growing. Getting more grants to expand on the ideas. I mean, we've already been successful to some degree in the convoluted way. We have a number of applied grants, Shams the most recent with Finding Pheno, but of course, Morten and Anton have been very successful with some applied grants that are very much based on the framework that came out of it. And we can look at the timing, but this is the way I will spin it to whichever funder I need to impress that they're giving us money for the right thing. And I anticipate more grants are welcome to build and build and build, whether at the PhD or the postdoc level or the new PI level. And I say we also need to get a degree of notoriety. We need to be as famous in the area of evolutionary hologenomics as people elsewhere, right? Because, and there are 2 reasons for this. Firstly, because I think we're right, and we need to persuade people we're right. There is a reason why I believe we're crusaders. I think, I think people, when they hear this, will realize, you know what, shit, maybe we should think about things this way. And they will only do that if they hear about what we're doing. And of course, that's important by doing papers. So we have published some of the first hologenomic papers, some that predated the center starting, like Liz's vampire paper that kind of got us there. A number of review papers, both some that came from just before it and also very recently. I mean, huge kudos to Lasse, Anton and team for putting out this really nice opinion piece about integrating holomics. And these are critical because we know these work. We've been contacted by people elsewhere who've seen these papers and want to join us, want to collaborate with us, and it really puts us as the founders of a massive discipline. This is why it's such an exciting time. Of course, we might be wrong, in which case it's awkward, but we will still learn an awful lot. But if we're right, we are the founders, the 4 founders of an area that will be very influential.